Hi everyone, our topic is about genomic analysis that has three different parts, which is the whole genome sequencing and sequence analysis, the comparative genome analysis, and the transcriptomics and proteomics. Genomic analysis is the study and examination of an organism's entire genome, which is the complete set of genomic material, including genes and non-coding regions of DNA. This analysis involves investigating the structure, function, variations, and interactions of the gene within an organism's DNA. Whole genome sequencing and DNA sequence analysis. Whole genome sequencing. It is a comprehensive process that involves determining the complete DNA sequence of an organism, enabling the identification of genetic variations and contributing to various fields, including medicine and evolutionary biology. DNA sequence analysis is a computational approach to interpreting the information contained in DNA sequences, involving processes such as sequence alignment, variant calling, and functional annotation. So as what we can see in the whole genome sequencing, the entirety of our genome data including both coding and non-coding DNA, as its name suggests, this type of genetic testing can identify variations in any part of our genome. And its main objective is to identify whether there are any mutations in our body that could cause health problems and provide information about our genealogy, which can be fascinating on a personal level but also helpful when it comes to determining our risk for a certain diseases. DNA sequence analysis is the databases containing genomic sequence data, which includes information at the level of the untranslated sequence, introns and exons, mRNA, cDNA, and translations. Here are the features of DNA sequence analysis: detecting open reading frame or ORF, initial codon which is ATG, stop codon which are the TGA, TAA, and TAG. These two several features may be used as indicators for potential protein coding regions in DNA. Sufficient ORF length, recognition of flanking Kozak sequence, pattern of codon usage, a general preference for GC over AT in the third base position of a codon, ribosome binding sites, and alignment with the homologous protein sequences.
Comparative genomic analysis.
Genomics is biological research field that compares genome sequence of various species, allowing researchers to understand molecular difference and evolutionary change among organisms. It helps identify conserved genes and unique characteristics in each organism. How are genomes compared? Comparing genomes features like size, number of genes, and chromosomes numbers allows for comparative genomic analysis. For instance, Arabidopsis thaliana, despite having a small genome, has nearly twice as many genes as Drosophila melanogaster, demonstrating that genome size doesn't correlate with evolutionary status or genes number. Genome comparisons involve analyzing the genetic materials of different organisms to identify the similarities, difference, and relationship. Comparative genomics as sequencing technology advances called benefits animal and ecological studies, agriculture, biotechnology, and zoology by revealing subtle difference between species, ranging evolutionary understanding, and identifying new conservation strategy for rare and endangered species.
Transcriptomics and proteomics.
Transcriptomics is the study of transcriptome, a term now widely understood to mean the complete set of all the ribonucleic acid or RNA molecule expressed in some given entity such as cell, tissue, or organism. Transcriptome is the set of all transcript in a given type of cells. In other words, it is a snapshot of expression profile at a given time of cell. In transcriptome, they are not only the classical messenger RNAs coding for protein but also microRNAs, long non-coding RNA, and other non-coding RNAs recently discovered that do not code for proteins. These RNA transcripts cooperate to regulate cell growth, development, apoptosis, and many other important physiological processes. What are the different types of transcriptomics? Initially, transcriptomes were analyzed and studied using express sequence tags, libraries, and serial and cap analysis of genes expression. Currently, the two main transcriptomics techniques include DNA microarrays and RNA sequence. The DNA microarray is a tool used to determine whether the DNA from a particular individuals contain a mutation in genes like BRCA1 and BRCA2. The chip consists of a small glass plate encased in plastic. Some companies manufacture microarrays using methods similar to those used to make computer microchips. RNA sequence is a sequencing technique that uses next-generation sequencing to reveal the presence of quantity of RNA in a biological sample, representing an aggregated snapshot of the cell's dynamic pool of RNA, also known as transcriptome.
Proteomics is the study of the interactions, functions, composition, and structures of protein and their cellular activities. Proteomics provides a better understanding of the structure and function of the organism than genomics. Proteomics is used to investigate when and where proteins are expressed, age of protein production, degradation, and steady-state abundance. How proteins are modified. Example: post-translational modifications, such as phosphorylation, the movement of proteins between subcellular compartments. Post-translational modification provides the rapid connection of transcriptional programs, including transcription factors and signaling proteins. It is vital that plants quickly respond to changes in the environment in order to survive under stressful situations. The different types of proteomics in genomics: expression proteomics, functional proteomics, and structural proteomics. These three are the major types of proteomics. Expression proteomics experiments are usually used to study the patterns of protein expression in different cells. For example, a tumor tissue sample is compared to a normal tissue sample to identify differences in the levels of proteins. Functional proteomics is one key tool scientists use to reveal the roles of proteins. For example, functional proteomics applications are already showing how the tau protein is involved in Alzheimer's disease and how specific proteins increase cancer risks. Structural proteomics is the application of protein chemistry and modern mass spectrometric techniques to problems such as the characterization of protein structure and assemblies, the detailed determination of protein-protein interactions. Example of structural proteomics was the recent analysis of the nuclear pore complex isolation of specific subcellular organelles or protein complex by purification can greatly simplify the proteomic analysis.